So let me start this lecture with an example. This is Kate Miller. She's 55 years old, and she normally feels very well, but for a couple of weeks now, she feels palpitations every now and then. She feels tired during the day, doesn't sleep well, goes to see her GP, and he refers her to a cardiologist. And her cardiologist, she recommends recording a long-term ECG, an electrocardiogram, a so-called Holter ECG, which analyzes or records the heart rhythm for 24 hours every day for a couple of days. And then this signal, this long-term signal, can be analyzed. And yeah, her cardiologist says the diagnosis is quite clear. Kate Miller suffers from atrial fibrillation. And the question now for you maybe is, what is fibrillation? And the question for her is how can we treat this, and how can we avoid any negative consequences of atrial fibrillation, which can be quite severe. So atrial fibrillation is a main cause of stroke, and stroke, of course, is a very severe complication. But atrial fibrillation also increases the risk for heart failure and death in general quite dramatically. So what is atrial fibrillation? The first aspect in this term is atrial, and this refers to the part of our heart. So our heart is here split in four chambers, the two main chambers, but we also have the two atria, the left and the right atrium. And those four chambers, normally in the healthy case, undergo a very coordinated and synchronized activity. So we have a natural pacemaker up here that drives every single heartbeat. And then we have a coordinated activation of the atria. The activation focuses here at this junction between the atria and the ventricles, so the upper and the lower chambers. And then the lower chambers are activated in a very synchronized way to ensure efficient pumping of the blood into the body. This is how it should look like and how it normally looks like. In the case of atrial fibrillation, this looks quite different, especially in the atria, in these upper chambers. So here we don't have this clear start of an activation and then an end of the activation, but we have self-perpetuating so-called reentrant activation that is very chaotic, that does not allow for efficient pumping function, and that also leads to a rather irregular conduction of this electrical activity to the lower chambers of the heart. So that's atrial fibrillation. How is it normally treated? Well, there are some drugs that can be tried to terminate this reentrant arrhythmia, that can also try to prevent new episodes. But the only curative treatment is to actually insert a couple of catheters inside the heart. So the clinicians enter the body through one of the veins at the leg, and then advance a couple of catheters inside the heart. So we end up here in the right atrium, and then they puncture the septum between the two atria and can enter the left atrium, which is in most of the patients the source of the arrhythmia. And the default treatment is then to isolate the orifices of the pulmonary veins, so the blood vessels that bring back the blood from the lungs. And we have two of them on the left, on one side of the left atrium, and two more on the septal side of the left atrium. And then some form of energy is applied. This can be radio frequency currents. This can also be cold or pulsed electrical fields that just render this tissue non-conductive. So the clinicians introduce some scarring into the atria on purpose to basically isolate those triggers of the reentrant arrhythmia from the rest of the atrium, and by this render them invulnerable. So this procedure looks something like that. They have a lot of monitors They maneuver the catheters, record the signals, and then apply the energy in the end. And this pulmonary vein isolation is quite successful in many of the patients. So they have atrial fibrillation before, then the veins are isolated, indicated here by these black bands that separate the pulmonary veins from the rest of the atrium. And then if everything works well, atrial fibrillation stops. And this works well in around half of the patients. So for them, really the problem is solved. They don't experience any further episodes of this arrhythmia. They don't feel the palpitations anymore. They don't feel so tired during the day, and their increased risk for stroke and heart failure is ameliorated. But there is this other half of the patients where this doesn't work. And of course, it would be great both for the patient, but also for the treating clinician, to have some information before if this intervention will likely be successful for a specific patient or not. And this is exactly the question with which our clinical collaborators here in Karlsruhe and Freiburg approached us and asked us, well, we have ECGs, right? We do record the electrical activity during atrial fibrillation. Couldn't you use some magic machine learning on that to predict whether this pulmonary vein isolation, or in short PVI, will likely be successful or not? And we thought about it and said, okay, well, this is a time series signal. This is just one lead, but normally we have multi-lead recordings. And we are interested in a binary outcome, either it is successful or not. So this seems like a pretty well-suited task for a machine learning classifier. So we said, okay, please provide us with the data, and we'll try to come up with a classifier and train it. So they went back into their clinical databases and checked for how many patients they do have such a recording during AF, for how many they also know whether it terminated or not, and for how many patients they would have consent to use their data for such a research project. And then they came back and said, okay, they found between 40 and 50 patients. And we said, well, that's not bad, but this is clearly not enough to both train and then evaluate or test such a classifier. So we were a bit disappointed and thought, oh, well, maybe we cannot really do that. And went back, but gave it a second thought and thought, well, maybe math could help here. Because what we in my group at KIT's Institute of Biomedical Engineering do as our core business, I would say, is modeling the function of the heart, and especially this electrophysiological function, so everything that's related to the heart rhythm, by mathematical models and doing computational modeling and simulation with that. And the way we approach this is that we first analyze this biological system. So our heart is really a multi-scale system. And I think it also helps to understand it as a multi-scale system to then represent its function with mathematical equations that can be the basis for in silico simulation studies. So the lowest level that we consider in our models are single ion channels in the membrane of a cell. So those are really just a few nanometers wide, and they control whether an electrically charged particle, an ion, can move from the inside to the cell to the outside or vice versa. And those ion channels can be analyzed with experimental techniques. Those ion channels can also be modulated by drugs, as we see here, for example. Now a drug binds, the channel opens, and then the ions can flow through it. And this flow of these electrically charged particles can be measured as a current. So there are established experimental techniques like voltage clamp or patch clamp measurements, where the behavior of such channels in the membrane can be characterized quite well experimentally in biological experiments in a petri dish, for example. And then we have multiple of those channels inside one muscle cell of the heart. So this is a cardiomyocyte, and we see the cell membrane. We also see this one type of channel, but we also see that there are multiple other channels, and they all interact with each other. So there are channels that are mostly responsible for depolarizing the cell. There are also repolarizing currents, and there is a lot of ions being moved around from inside to the outside, but also inside intracellular calcium storage, for example, with every activation cycle of the heart, so with every heartbeat. And this combination of the different channels gives rise to what's called the action potential. So here we see the electrical voltage between inside and outside of the cell across time. And it's something that, yeah, some of you might still be familiar with from neurons from high school biology. So that's the single cell level. And then in the heart, many of those cells are electrically connected. They form a functional syncytium. And we can, of course, also build tissue and organ models. So this is a model of the two atria. Here we look into the one of the pulmonary vein orifices. And this is the right atrium here. And the electrical excitation then spreads across this functional syncytium, across this tissue, because all of those cells are electrically coupled and communicate with each other. And last but not least, this heart is also immersed in our body, in our torso. And the spatial gradients of the transmembrane voltage that we just saw here, they are the sources for an electric field that extends everywhere in space and also in the body. And this is what we can measure on the body surface as an electrocardiogram. So now we moved all the way from the nanometer scale via the micrometer and centimeter scale, almost up to the meter scale. So this is really a multi-scale system. And the way we approach this is then to represent each of those scales with adequate mathematical equations. So the opening and closing of those ion channels can be represented by ordinary differential equations that describe the time evolution of the open probability. Then on the cell level, where we have those multiple connected and interacting channels, those are, from an engineering point of view, just a parallel circuit in the cell membrane with the capacitive behavior of the cell membrane itself. If we think about excitation spread on the organ level, this is clearly a spatial process. So to describe this, we need to move from ordinary differential equations to some partial differential equations that also represent this spatial dependency and the spatial spread of excitation. And last but not least, the electrical signal on the body surface can be computed by solving Poisson's equation with this spatial gradient of the transmembrane voltages as the source term. And then, of course, we link all those models across the scales so that we can introduce a change in an ion channel, for example, because one channel is affected by a specific gene mutation, or because we apply a drug that affects the behavior of some of those channels, which affects the currents and voltages here, and then can be translated to changes in parameters of those ODE models. And then we can analyze this effect on the ion channel, or of the effect of this change on the ion channel level on behavior on the cell level, on the tissue level, and also on the ECG in a closed chain of experimental settings that are all linked to one another. So that's kind of the workhorse we will be considering here as one representative of such mechanistic physics-based models. So the equations here really represent the basic laws of physics, and the parameters that are here represent what we know about physiology and about how our body functions from a lot of experiments. So this is what's meant by mechanistic. Two thoughts on model philosophy. I brought two quotes here, and the first one is, the best model of a cat is a cat, preferably the same cat. And then there is another one by George Box, who said, all models are wrong, but some are useful. And I agree much more with George Box here, because what I want to show you is to be useful in the setting we're discussing here, a model needs to be a simplification of the real world entity. We need to abstract things. We need to deliberately focus on some aspects and thereby neglect others to gain something by using those models. So how can we use them? Well, the nice thing is that we can change model parameters on all those different scales. So as mentioned already, ion channels can be affected by drugs. They can be affected by gene mutations. And there are also influencing factors on the cell level. So if you have a chronic disease, for example, the protein expression of those ion channels will adapt to the change in the environment. Also, if you have, for example, a kidney disease and your body is no longer able to maintain a balance of the different electrolytes like potassium, then those will change and will, of course, also change the behavior of the model. On a tissue level, it's obvious that we can represent different shapes of the heart, different sizes, but also changes in the tissue microstructure. So there is a lot of diseases, including atrial fibrillation, that lead to fibrotic infiltrations that change the electrical conductivity locally. And this can be represented here as well. And on the body level, of course, the individual shape, maybe also changes in the electrical conductivity of the lung in response to certain physiological alterations. Once we have parameterized the model, so all those arrows up here are input parameters, we can run a simulation and then extract outputs of the model on all those levels as well. And those are readouts of the physiological function of the heart here. And in this case, it's electrophysiology. So we can look at ion currents on the single current level. We can look at the action potential and cellular arrhythmia markers on the cell level. On the tissue level, we can see whether we can actually induce such a reentrant arrhythmia, such as self-perpetuating chaotic excitation. And of course, on the body surface level, we can derive a virtual ECG. And what's obvious from here is that on the body surface level, we can simulate signals that we can also easily acquire in real life. So recording an ECG is cheap, it's non-invasive, it's done thousands of times every day. Also on the ion channel level, we can assess this rather easily with experimental techniques. But the cell level and the tissue level is much more difficult to assess experimentally, especially in a human setting. So this is where the models can help also to bridge those findings from one level to another and provide this closed chain. So to summarize this, computer modeling and simulation can serve as a very controlled research environment to help us understand fundamental physiology and also how things can go wrong, so pathomechanisms. The models can help to evaluate diagnostic and therapeutic approaches. And these are often called in silico studies, in contrast to in vitro or in vivo studies, which would refer to experiments in a dish or in an animal or a human. And of course, this can also be used as a tool to optimize medical devices, for example, to decide whether the case of a cardiac pacemaker should rather be flat or a bit thicker, and how this interacts with the electric field. This is a classic design optimization problem. This is one aspect, but then the second aspect is what I want to focus on today, is that it can serve as a scalable research environment to generate big and quality control data sets for machine learning. So we can run many simulations, and if those differ in output, we know they only differ in output because we change the parameters, we change the input. So there's a clear cause-effect relation, and we can rule out any other confounding factors like in a clinical study, whether the patients feel well, whether one patient came there during summer where it was 30 degrees hot, and the other one maybe at freezing cold temperatures. And this is a very controlled environment there as well. And last but not least, the third way how such modeling and simulation tools can lead to impact is this vision of personalizing medicine through digital twins, with which one refers to personalized models that represent the individual anatomy, but also function of one very specific person or patient. So let's come back to our problem here. We wanted to predict whether pulmonary vein isolation would be successful or not, and we saw that just by using real-world measured ECGs, we don't have enough to robustly train and then also independently evaluate such a classifier. But well, we do have this multiscale model. So what we could do is run a lot of simulations here on the organ level model, see whether we can induce an arrhythmia, then introduce the virtual intervention, the pulmonary vein isolation, and relate this with the body surface ECG. And this is what we did here. So such an episode of atrial fibrillation is what we see on the left side. The color codes for the transmembrane voltage, and we see this self-perpetuating re-entrant activation that's quite chaotic. We can also compute the body surface potentials for this and derive the virtual ECG. So this is what would be the input data for our classifier. But then we also need the ground truth labels. So will pulmonary vein isolation be successful or not? And then, of course, we can introduce the pulmonary vein isolation here in the model and see whether this terminates the arrhythmia or whether it doesn't terminate the arrhythmia. So those are the ground truth labels. In that study, we did that for more than 1,000 simulated episodes and could use that then to train a machine learning classifier. So we had those simulated ECGs together with the ground truth labels. Will the virtual intervention terminate this arrhythmia episode or not? And then could train a classifier to make this prediction based on the ECG and then compare that with the ground truth label. So this was done purely on those 1,000 synthetic data sets. A rather basic machine learning classifier, a decision tree with somewhat understandable features was trained. And once we were happy with the performance on the simulated data set, we froze the algorithm, froze the classifier, and then went back to the clinicians and said, okay, now we're ready. Give us those 40-something measured ECGs, and we'll feed them into the exact same algorithm that has been trained on the simulated data and see whether this can predict the true termination rate on those actual patients. And this worked surprisingly well with a specificity of more than 80 and a sensitivity of more than 70%, and highly consistent across multiple recordings of the same patients. So this can be a way for Kate Miller to inform the shared decision-making of whether she goes for pulmonary vein isolation, so this ablation procedure, or whether she will first try some antiarrhythmic drugs, or whether maybe something more than just pulmonary vein isolation needs to be done. And this can be valuable information to inform this shared decision-making between Kate Miller and her cardiologist. And this machine learning classifier could only be trained because of the simulated signals. So in that scenario, modeling and simulation, mechanistic physics-based bottom-up models were an enabling technology for the data-driven machine learning approach. And that's, I think, one example for some of today's challenges with machine learning for medicine. So let's zoom out a bit now from this specific example and look more at the big picture. And what we see often here are that we miss data sets of adequate size with high quality labels. So either we have big data, but they are not very well annotated, or we have very well annotated data, but in small numbers. And often there is, in theory, big data, but in practice, it boils down to very small data. So that's what I call the big data, but small data paradoxon in clinical data science, because there is a lot of data and it feels like we are drowning in data. But for specific applications, we are often facing very small data. Those data sets are then likely biased and unbalanced. So if we get data from a hospital, of course, this contains only patients that walked into a hospital and are not very representative for a general population often. Unbalanced in the sense that for specific pathologies, for specific events, we often have much bigger negative classes than the positive ones. And there can also be bias and imbalance in terms of age distribution, sex distribution, and those kinds of things that really put a risk on the fairness of the tools that one might develop here. So just as an example, one of the references down here, they look, for example, in race differences, and showed that something that's trained for European hearts might not work well for the average Asian heart, for example. So that's a problem. And then often we also have intransparent methodology, that something is trained, but the data are not shared. Sometimes not even the code or the weights are being shared. And specifically for data, this is often due to data privacy issues, that even if the researchers would be willing to take the effort to share those data, they might not be allowed to do so. And this, of course, poses risks to this fairness aspects with the capital letters. So F for findable, A for accessible, I for interoperable, and R for reproducible. And maybe synthetic data could help us to fight some of those challenges. Maybe we could overcome this big data but small data paradoxon. Maybe we could work towards more balanced, more and less biased data sets, and we could maybe generate data that are easier to share later on. So what are synthetic data specifically? So there is a definition by the Royal Society and the Alan Turing Institute who say that that's data that has been generated using a purpose-built mathematical model or algorithm with the aim of solving a set of data science tasks. So someone builds a mathematical model, and that can be a mechanistic model, or it can also be a data-driven model itself. That should produce specific data that will later on be used to solve other data science tasks. If we look at the number of publications on PubMed, for example, that mention synthetic data, this is clearly increasing. Most of the synthetic data sets are in the imaging field, and some are also for tabular data. And the hope is that those data could reduce cost and time, and also help fighting rare diseases and conditions. The hope is that synthetic data might enhance the predictive power of AI models, that they contribute to more fair treatment and more fair treatment recommendations also across diverse patient populations, and that they facilitate easier access for researchers to high quality representative multimodal data sets without exposing sensitive patient information. So how do those synthetic data look like? Well, we've seen the ECGs before already, but there are many more application fields. So up here, we, for example, see some avatars, and on the top row, it's always the real data. On the bottom row, it's the synthetic ones. So here they look somewhat similar, but clearly not the same. The same here on panel C with the MRI images of brain tumors. The ones that are maybe hardest to discern and to distinguish between synthetic and real are those histology slices, which are stained images of human skin tissue. And of course, ECGs are another way here. How can we generate such synthetic data? Well, we've seen one example already, which was a physical model, or what I would call a mechanistic bottom-up model that was used here to train this predictor for pulmonary vein success. But that's not the only way. There could also be statistical models like GANs, for example, generative adversarial networks, or also with newer generative AI methods, purely data-driven, generating new data sets. But of course, there it's less obvious how the input and output relate to one another and what should be the label for this newly generated data set. But it's a big group of models as well that are working purely on data-driven statistical models to generate new synthetic data. And then they can also be combined in hybrid models that have some blocks that are purely statistical and some blocks that really consider the physics and check, for example, that the data that are generated fulfill basic equations that are known that should hold true. And this could, for example, be a physics-informed neural network combined with some other data-driven methods. So that's for the simulator to generate the synthetic data. And then when we want to apply them for machine learning, the training can either be done on real data only. Then, of course, we wouldn't even need a simulator. It could be done on synthetic data, or something that's often called hybrid machine learning or hybrid data sets, where in the training data, the synthetic and the real data are combined already. And the same holds true for testing. So once the algorithm has been trained, has been frozen, and a final evaluation is done, this should ideally be done on real data, but it's also good to keep in mind that this could also be done on synthetic data or on hybrid data as well. So can those synthetic in silico data help us to overcome those challenges? Well, we can keep in mind that especially the mechanistic models, they are very controllable. We have a clear relation between input and output, and we see the output changes. We know this is the case because we changed some of the input parameters. So there are no uncontrolled confounding factors. The results they deliver are inherently quantitative, and the spatial and temporal resolution is basically only limited by how much time we want to spend on the computation. They are mechanistic, so they are explainable in a way and understandable. They are personalizable, but they are not personal per se. So we can have a mechanistic model that does not contain any sensitive patient-specific information. So could that help us, or could that help Kate Miller? So let's come back to her and consider what happens if our previously trained classifier would say very likely pulmonary vein isolation will not be sufficient for her and will not cure her atrial fibrillation. Or what happens if she got a PVI already and comes back with a recurrent arrhythmia? What could be done then? Well, let me present you a vision for this way of impact using personalized medicine and digital twins. So together with her cardiologist, they could decide that she undergoes medical imaging, cardiac MRI. Then from those data sets, a computational model of her individual heart could be built. And then different ablation strategies and therapeutic options could be tested in a dry run on this computational platform before then implementing one of them in the clinical intervention. And to somehow organize this in a more systematic way, I find this concept of ensemble simulation spaces quite helpful, where we could say, okay, if we want to build a model of not just a heart, but of Kate Miller's heart, what do we need to consider? Well, there is obviously the anatomical dimension. So the shapes of our bodies and also of our hearts differ, so we need to consider that. But that's not enough. Also, the function differs. So while your heart and my heart might look a bit different, also the speed with which an excitation and activation travels across our hearts can be different. So it's not enough to personalize anatomy, we also need to personalize function. And if we find ways to do both of this and locate Kate Miller on both of these axes, then we have one point in this two-dimensional space, and that's her digital twin. How can that look like? Well, for an anatomical personalization, this is basically a solved problem. So we have cardiac imaging, we get those volumetric data sets. They can be segmented to identify the hearts or the atria, and then build a computational mesh representing the individual anatomy of Kate Miller. We can then also augment these meshes with some population level a priori information. So we know that atria are composed of different structures. So we have the ring where the mitral valve is here, for example. We have also certain fiber alignment that can be annotated on those personalized anatomical meshes to augment the data set. Functional personalization is actually often the more challenging and more tricky part of building a digital twin. So as mentioned, we would need to for example, need to know how fast an excitation travels. And we could get a very first guess, a global measure of how fast the wave travels by looking at the ECG and see how wide those different waves are, and it can give us some information. If that's not enough, if we're not happy with such a global information for our specific use case, then we might need to have a more spatially resolved information. And this is something that's routinely acquired during the intervention. So what we see here is a map of the left atrium, and the color basically says at what time this part of the atrium was electrically activated. So we have a wave that travels from the blue regions via the green ones all the way to the red. And that, of course, can also be used to derive information about the conduction velocity of that activation. So that helps us to build Kate Miller's digital twin. And then we want to do something with her digital twin. And that's why we add this third axis here that's called the intervention envelope. So we want to take her digital twin and then move it along this third axis to try different therapeutic options. And this could either be different ablation strategies or different drugs as well, if we have the mechanism of action of this drug included in our model. And as mentioned at the very beginning, we can represent the action of the drugs on the ion channel models. But let's focus on ablation now. So we know that pulmonary vein isolation alone will likely not be sufficient for her. She belongs to this 50% of the population where atrial fibrillation cannot be cured by only isolating the pulmonary veins. What else could be done in this field of personalized medicine? Well, if you look in the clinical literature, also if you talk to clinicians, there are many more ablation strategies. So pulmonary vein isolation is always the first step. It's the cornerstone. But there are other anatomical options. So one could add, for example, here a line at the roof of the left atrium and some additional line here at the mitral isthmus. One could look at the substrate. So how diseased is the atrial tissue itself? If the problem is not only coming from the pulmonary veins, but there is also a problem in the body of the left atrium itself. This can be assessed by looking at the voltage of the electrical signals that are acquired during the intervention. And if the voltage, the amplitude is low, then this is considered to be non-healthy tissue that might be good to ablate. And there are many clinical trials that follow this route. One could also look at the conduction velocity and target slow conducting areas. And one very attractive option is also to get a readout of the state of the substrate before the intervention, before inserting the catheter into the heart by another MR sequence that uses a contrast agent that highlights areas that are supposed to have this fibrotic infiltrations. So all those are also being tried clinically. And if you ask, this is something that has been done, many clinicians, they will tell you very different things about what's the best approach. And empirically, it has been shown that none of them works for the majority of those patients. So another option could also be to build this digital twin, then induce such a reentrant arrhythmia, and target the organizing centers of those in the digital twin. And this is what we call functional ablation here. So it's not anatomical, it's not substrate-based, but one actually tries to induce the arrhythmia. And for Kate Miller, the question is now, which of those strategies is optimal? And if you're faced with such design questions in a technical, in an engineering setting, like for example, which of those slightly different versions of this car is aerodynamically more optimal? Well, what one would normally do is to run a simulation study and to assess the different options in a computer experiment before trying that and building that car. And this is what we can now also do with Kate Miller's digital twin. So we have the anatomical personalization. We looked into how we can functionally personalize it. And then we can run a virtual stress test. So we can really try to induce many atrial fibrillation episodes in her personalized computer model in her digital twin. And if that's successful, it looks something like we've seen before. And then we can try different interventions. So adding those non-conductive ablation scars to her model, to her digital twin, and then see whether a specific ablation strategy terminates the arrhythmias that could be induced. And very importantly, we can also try whether once this intervention has been applied, whether it can be, whether we can still induce a new episode. And if that's the case, then maybe we should add an additional ablation. And that's what we did in a cohort of 29 patients. We built digital twins for all of them, and then tried all those different ablation strategies that are proposed in literature and applied every day in clinics on this cohort of 29 patients, which then showed very different rates of success. So the height of the bars here shows in how many percent of the cases this was successful. Green means complete termination, and red means it didn't terminate the arrhythmia, but it converted into a more organized form, less chaotic, that's supposed to be much easier to treat. So that would be considered a success clinically as well. And we can see that this is quite variable. But even with those strategies that work well, we are left with around 12% that don't convert And it's clear that there is no single strategy that will work well in all of the patients. So that's another way how we might be able to help Kate Miller by doing those in silico trials. So yeah, building not just one digital twin, but a whole cohort of many such digital twins that can then all undergo the same interventions and allows for comparing those different therapeutic strategies in a very systematic and controlled manner. That's nice, but it's not very scalable in two ways. One aspect is the computational time that we need to spend for each of those virtual interventions and then checking again whether it's successful, whether it was successful or not. And the other aspect in which this is not very scalable is, yeah, we spent quite a bit of effort to build those 29 models and then run a virtual trial with 29 patients. But if you want to do this with a few hundred, a few thousand, this is not really feasible. So what about computational time? Building such a model takes a few hours. Then you can fill a supercomputer for a couple of hours to then predict the outcome, which can be done before the intervention, not a problem, but it's not really feasible during an intervention that lasts for normally around 2 to 3 hours. So that's a time frame that's too short. So maybe we could use some machine learning to speed up those simulations and kind of learn a surrogate model that predicts the outcome of the simulation. And this is something that we tried recently for exactly this use case for predicting AF ablation success. So again, a synergy between mechanistic models that are up here, where we generate a model, run the simulation, and data-driven that tries to predict directly from the data what the outcome of the simulation would be. And yeah, this is not working perfectly yet. So if we look at this receiver operating characteristic curve, we would like this to be almost a rectangle reaching up to the top left corner. But here we have an area under the curve of around 0.75. The nice thing here is that we can really also provide a heat map for inducibility. So if we have such an atrial model, the classifier will not only say this is inducible or not, but for every point, it can say how likely it will be to induce there. And what we can already do with this is to say, okay, in those dark blue areas, it's very, very unlikely to induce something here. So we can save the computation time and do not simulate here. So that's one way of how this can be sped up and how we can address scalability in terms of computation time. The other scalability limitation I mentioned was just the size of this. So here we really built 29 models of persons that exist in real world where we had access to the data. But wouldn't it be nice if we could also move along those two axes, the anatomical and the functional axis, more freely, while still maintaining the anatomical and physiological plausibility? And this is what's often called a digital chimera then. So we would generate cohorts of models that don't represent individual persons, but the overall cohort matches very well the statistical properties of the real world cohort. So of our population in a specific city or in a country or a group with a specific disease, for example. And the good news is that this is possible. So on the anatomical envelope, we can, for example, use statistical shape models that encode the shape variability of a specific object in a population. So here we see different body models, different heart models. And if we build a statistical shape model out of these, we will get a mean shape, so a mean body and a mean heart, but also modes of variation. And those modes of variation are statistically coherent with the distribution that is seen in the real world. And then by varying those coefficients, Ri here systematically, we can derive new anatomical heart models that are realistic, but not derived from a specific individual. So we can get many of those hearts, and this allows us to scale up from 29 to 800 virtual patients. That's the work that was done in collaboration with Oxford University, where we really replicated clinical trials that were partly completed already, partly still ongoing, and could show that with those 800 virtual models, we could predict the outcome of the clinical trial success rate up to a few percent. So this is a very very strong testament of the validity of those models. But we could not only replicate things, we could also analyze why this was successful and why the different strategies were successful in some patients and not in others in a very systematic way. And by doing this, one can come up with such a decision algorithm for treatment selection that could be very easily translated into clinical practice. Because we saw, for example, that it's the size of the atria that matters. So if you have a big right atrium and a big left atrium, you will need more extensive ablation than if you have small atria. If just one of your atria is enlarged, different strategies are much more likely to succeed. And this is something that can be assessed easily clinically, that can also be implemented easily clinically, because once we have derived such a decision algorithm, which are frequently used in clinical practice, the physician, the clinician, does not need a simulation model at hand anymore. They can just follow through this decision tree, and in this way, help finding the optimal treatment for Kate Miller together with Loewe. So that's another way how we can generate cohorts for such in silico trials in a much more scalable way, and also with no direct connection to specific individuals anymore, that would allow sharing those data much more easily than sharing digital twin models. So what else can this be used for? We recently published a large-scale ECG data set that's called MetalCare XL, where we did exactly that. So we had multiple atrial models, multiple ventricular models, then varied not only the anatomy, but also the function within the range of parameters that are known to be plausible from literature. And then we could simulate the ECGs and add very controlled levels of noise, add very controlled levels of beat-to-beat variability, but also add specific pathology, specific diseases to those models, and then derive a cohort with not only healthy ECGs, but also some very specific for myocardial infarction, for example. And then, of course, those models need to be validated. And this is something we did here on the one hand by comparing the features of those ECGs with features derived from measured signals. And you can see that some of those, such as the amplitude of the P wave, match quite well, while others, such as the amplitude of the T wave, for example, still show some deviations. So this is one way how one can assess the validity of this ECG simulator. Another thing was that we showed this to clinicians, the simulated signals, together with measured signals, and asked them to guess whether it's simulated or not. This is what we call the clinical Turing test. And the very well-trained clinicians could identify most of the signals, but not all of them. And this is shown here in this classification matrix. And the nice thing about that is that this is a purely synthetic data set. So we could easily publish this, all 16,000, almost 17,000 ECGs, with full traceability. So together with all the model parameters that were used to run the simulation, so you know exactly why a signal looks in a certain way, because you can trace it back to a difference in the conduction velocity, for example, with which an activation travels across the atria here. So this is a research resource that was enabled by computer modeling and simulation. Some more use cases. This is, for example, the localization of ectopic beats. So we already heard about the sinus rhythm. That's the normal case where excitation travels from the atria to the ventricles. But there are also, or there can also be ectopic foci in the ventricles where an excitation is triggered here. And then the ECG looks something like this. And often this is completely harmless, but in quite a number of patients, this can trigger life-threatening arrhythmias, sudden cardiac death. And if the number and frequency of those ectopic beats becomes too high, then they need to be treated. The normal case is that, again, a catheter is inserted, and then the clinicians spend often hours locating the exact point where those ectopic beats originate from. And then they ablate it also in the ventricles. So they apply radio frequency current here to destroy the tissue and add a scar. And the question we asked here, can we implement this with a deep learning approach for non-invasive localization? So we only have this ECG. Can we predict where in the heart this beat originated from? And we designed such an in silico pipeline again. So many anatomical models, as we see here. In this case, it was 1,000 different anatomies, but we can also rotate and translate the heart inside the torso to capture the variability here, and then compute the ECGs and train a regression algorithm that would predict the location of this ectopic beat in the ventricles, and that worked quite well. So what we see here on the y-axis is the error in localization, and the first four are the machine learning classifiers, whereas the two on the right are what's currently considered the state of the art by solving this inverse problem with standard regularization techniques. So we could reduce this uncertainty in localization from a few centimeters to just a few millimeters, and that could help quite dramatically to tell the clinicians where to look first if they are after those ectopic foci that they want to ablate. In the scope of the atria, just to close, three more applications that were enabled by synthetic data. So one is that we can estimate how much of the tissue in the left atrium is affected by those fibrotic infiltrations. And this is a very valuable information for stratification. So to say, how high is the risk? Also, how high is the probability that a certain treatment will work? And this was trained purely on synthetic data and enabled an estimation of the volume fraction with an error of less than 10%. This is a study where we used a hybrid data set. So we wanted to identify, based on the ECG, whether the left atrium is enlarged or not. And there were quite a number of measured data, but we could show that by adding additional simulated data to the training set, we could increase the accuracy on the purely clinical test set. So all the reported performance metrics here are always on clinical test sets. And last but not least, there is another atrial arrhythmia, which is not atrial fibrillation, but atrial flutter. So this is this more organized form. where it would be interesting for clinicians to know before they perform the procedure which specific subtype it is, so around which anatomical obstacle this excitation is traveling, for example. And here also we could use such a hybrid data set to increase the accuracy on a clinical test set quite dramatically. In this case, we also tried training purely on simulated data, but this did not work well. So the domain gap between the simulated and the real world data was apparently higher for this pathology of atrial flutter than it was for atrial fibrillation, where a purely synthetic training set served the purpose. So those are some success stories, I would say, but there's also a word of warning. So this is a recent study that was published in Nature that showed that if you recursively train AI models on synthetically generated data, then they collapse quite fast. And what they mean by collapse is that the distribution of the data points converges to much narrower distributions. So while in generation 0, there was quite a wide distribution, then we lose those tails of the distribution once we move from generation 0 to 1 to 2 to 3 to 5 and so on. So this is something to keep in mind. I don't think it's the case for those mechanistic models, because here we don't recursively generate data. We use an established, well-understood model to generate one set of data. But then we don't use an AI model to generate a next generation and the next generation. But this is something that could happen, of course, with those statistical simulators. And it's something to keep in mind. And that was elegantly shown here in this publication for a number of use cases. So this seems to be a quite universal phenomenon and not limited to specific approaches. So in conclusion, I hope I could show you some examples of synergies, how simulations can help to increase data set size and quality. We can augment on additional levels. So for example, mixing hearts from one patient with a torso from another one, do this mix and match, work towards more balanced classes, just running a balanced number of simulations for each classes. We have access to ground truth labels with very high certainty. And it can be a way to contribute also to the 3 R's of animal testing. So to replace, reduce, and refine animal testing by doing some aspects of this in silico first, and then only testing the most promising approaches in an animal model, maybe also being able to reduce the number of animals there. Of course, there are also challenges. So what we've seen is that we need to cover the relevant variability. This is true for purely data-driven models, but this is also true for mechanistic simulation models. So if we have a statistical shape model that was trained only on, let's say, young men in Germany, then we should not expect the statistical shape model or the instances derived from them to be representative of, say, old women in the US. So it's important to trace this variability along the full chain of data generation, but also of simulation. And as I showed you on the last slide, there can also be domain gaps between simulated and real data. And there are specific methods also to bridge this domain gap or to close it, to at least make it smaller. And you find it in the literature as sim to real. So I think indeed synthetic data can help to reduce the cost and time required for clinical trials, especially for rare diseases and conditions. It can enhance the predictive power of certain AI models in personalized medicine. It can also contribute to the delivery of fair treatment recommendations across diverse patient populations. And it can enable researchers to access high quality, representative, multimodal data sets without exposing sensitive patient information. A few examples of those data sets are just shown here. So I mentioned the ECGs already, but we also published this shape model and instances of it together with some data for this project of localizing the origin of the ventricular extrabeats. Those were actually 1.8 million simulated ECGs. They are also openly available. The same holds for the simulation tools. So for cardiac electrophysiology, most of the work is based on the OpenCarb simulator, which is free for academic use. It comes with a lot of training and documentation material. It also comes with a list of community resources, so anatomical models, data sets that can be used, also pre- and post-processing software that works well with the simulator. So it's something that should in theory be accessible for many researchers and try to reduce redundancy and live up to these ideas of fair and open science. With that, I don't want to miss to thank a lot of people who have contributed to this, also funding bodies who enable this research. And would like to close this lecture on this synergy between mechanistic and data-driven models. And I'm looking forward to the discussion now.